Now, the next speaker was supposed to be Till Bergman. Till was not able to make it for today, but Max kindly accepted to kind of present Till's talk. So, Max, are you with us right now?
I am. Can you hear me?
Yes. Good afternoon.
Great. Hello. Thank you, Ahmed. Also, I'm going to briefly present our approach for individual functional connectivity and E-field optimized TMS targeting, why we developed this approach and how we are using it in a current TMS fMRI study. And to start with, let's first look again briefly at functional connectivity optimized TMS targeting, where the idea is, in contrast to the more commonly used heuristics for coil positioning, to personalize the TMS target and the coil position by identifying and then stimulating the subject-specific spot in, for example, the left DLPFC, whose resting state activity is most strongly correlated or anti-correlated with activity in a deeper region that we would like to indirectly modulate, for example, the subgenual cortex, or in the case of our study, the ventromedial prefrontal cortex, the VMPFC. However, this functional connectivity-based approach has several problems. First, the personalized target is most commonly or is defined by only a single voxel, and that means ignoring the rich spatial information coordinate commonly simply to the subject scalp to define the coil center position. And third, even though this gives us a relatively good idea about where to roughly place a coil, we basically still have no clue how to optimally orient the coil in a given individual. Next slide, please. And this is where E-field simulations come into play. Such simulations have, for example, shown that simply placing the coil directly above the cortical target will not necessarily guarantee optimal stimulation of that target. So using a grid search procedure and then simulating E-fields for numerous different coil positions and orientations, we can use these simulations to tell us not only where to optimally place the coil, but also how to optimally orient the coil in order to maximize the E-field within our identified cortical target. Again, this approach has a few problems. For example, most commonly, the E-field is only maximized within a very small region of interest surrounding the identified target coordinate. Again, ignoring the whole brain properties of the induced E-field and its spatial spread, basically, across the brain that clearly goes well beyond this small region of interest. And this can then result in a situation, as shown here on the slides, where if you really consider the whole connectivity map on the left, you have this relatively large cluster of negative functional connectivity that you would like to stimulate, and that is oriented as indicated by this dashed orange outline. And on the other hand, you have this induced E-field that results from the seemingly optimized coil position and orientation, and that is oriented quite differently. And this, of course, can lead to unwanted off-target effects where, for example, areas that you would rather not like to stimulate also experience high E-field strength. And this is why, next slide, please, we aimed to slightly extend this approach by taking into account the whole brain topography of both the individual functional connectivity map and the induced E-field. So within this approach, we not only maximize the E-field within the target cluster, but, and that's a crucial point, we also maximize the overlap, the whole brain overlap between functional connectivity map and induced E-field, thereby minimizing off-target effects and hopefully increasing the specificity of TMS. And the resulting E-field, as you can see, now much better aligns with or follows the orientation of this negative functional connectivity cluster on the left that we would like to target. And also, next slide, please, you can see that these two approaches would result in considerably different coil orientations. Next slide, please. At the moment, we're using this approach in a TMS fMRI study where, first of all, for all of our subjects, we generate an individual whole brain functional connectivity map based on multi-echo resting state fMRI data and a seed-based functional connectivity analysis using the VMPFC as our seed region. We specifically use multi-echo fMRI because it has been shown to not only have better SNR in regions we are particularly interested in, so for example the VMPFC, but also to result in considerably more reliable functional connectivity estimates at the single subject level with less amounts of data than, for example, conventional single echo sequences. Next slide, please. We then identify the largest clusters of negative and positive functional connectivity within the left dorsolateral and dorsomedial prefrontal cortex as our stimulation targets of interest. Next slide, please. And then centered around these clusters, we define a grid of many different coil positions and orientations, simulate TMS-induced E-fields for each combination, and then eventually we'll choose the coil position and orientation that simultaneously maximizes the E-field within the functional connectivity cluster of interest, and importantly, the overlap between E-field and the functional connectivity map. As you can see, the resulting E-field relatively nicely overlaps with our target cluster, with the corresponding coil position and orientation shown at the bottom. Next slide, please. And in this study, we not only define coil position and orientation based on E-field simulations, but we also use the simulations to individually determine the stimulation intensity, here aiming for a mean E-field strength of 100 volts per meter within the respective target cluster. And in addition, as you can see on the left now, we define for both of our functional connectivity targets of interest an active control target that ideally fulfills several conditions. First of all, it should be as close as possible to the actual target of interest in order to produce similar somatosensory and auditory effects. On the other hand, it should show as little functional connectivity with the visible, and it should induce an E-field in the target cluster of interest that is as weak as possible. Next slide, please. Then the resulting coil position and orientation and the stimulation intensity are then used in a concurrent TMS fMRI measurement to probe the TMS-induced bolt effects in both the directly stimulated area and the functionally connected regions across the brain. And for that, we use two of these ultra-thin MR surface coils to achieve whole brain coverage. And one of these coils is placed directly below the TMS coil, so basically between TMS coil and the subject's head to achieve higher SNR at the stimulation site. And then finally, next slide, please. Contrasting stimulation of the actual functional connectivity target of interest against stimulation of the control target will give us a subject-specific map of TMS-induced bolt modulations that are now specific for the functional connectivity target, where now for this pilot subject here, so this is single subject data, we see a positive bolt modulation within the directly stimulated DLPFC cluster, and then these negative bolt modulations in functionally connected regions, for example, the VMPFC, so the area we are interested in. And we hope that this will hold true and also work out in further subjects. And to come to an end, I think this slide also nicely sums up our approach for precise TMS targeting of functional brain networks and deeper brain regions in single subjects. And with that, next slide, please. I would like to thank you for your attention and also my supervisors and collaborators. And if you have any questions or comments, please feel free to reach out. Thank you.